Bismillahirrahmanirrahim. We have discussed acute inflammation. Now we are going to talk about outcomes of acute inflammation. As you can see in the picture, whenever injury is there either due to infection, bacterial infection, toxins or trauma, it leads to acute inflammation. Acute inflammation includes the vascular changes, neutrophil recruitment, and mediators are also secreted. If acute inflammation is not resolved, it leads to chronic inflammation. If the offending agent is not removed, or it may be present from the onset of injury, for example in viral infections or immune responses to self-antigen. So you can see in result of angiogenesis or mononuclear cell infiltrate or fibrosis, that is scar is seen while talking about the chronic inflammation. If the acute inflammation heals, if healing is seen, there is fibrosis, that is loss of function is seen over there. Acute inflammation can show resolution, that is regeneration and repair, when the injury is limited or short-lived, or when there has been no or minimal tissue damage, and when the injured tissue is capable of regenerating. The usual outcome is restoration to structure and functional normalcy. This involves neutralization, decay or enzymatic degradation of various chemical mediators, normalization of vascular permeability, and cessation of leukocyte emigration with subsequent death of extravasated neutrophils. So you can see resolution in the diagram, that is written clearance of injurious stimuli, clearance of mediators and acute inflammatory cells, replacement of injured cells, and normal function. Chronic inflammation can also occur due to injury, that is viral infection, chronic infections, persistent injury, and autoimmune disease. In chronic inflammation, there is written fibrosis, and in bracket it is mentioned scar. What do you mean by the term scar? Scarring is a type of repair after substantial tissue destruction, as in abscess formation, or when inflammation occurs in tissues that do not regenerate. As a result, in those tissues, it is filled by connective tissue, and in organs in which extensive connective tissue deposition occur, in attempt to heal the damage or as a consequence of chronic inflammation, the outcome is fibrosis, a process that can significantly compromise function. As you can see, with fibrosis it is written loss of function. So this is about the outcome of acute inflammation. Let's talk about the leukocyte induced tissue injury. Leukocytes are capable of secreting potentially harmful substances such as enzymes and reactive oxygen species that can lead to tissue injury. For example, as a normal attempt to clear the damage in dead tissue, for example after a myocardial infarction, in an infarct, inflammation may prolong and exacerbate the injurious consequences of ischemia, especially upon reperfusion. And another example include the autoimmune diseases or allergic diseases, including asthma. In allergic diseases, hosts react excessively against the non-toxic environmental substances. You know that in allergy, excessive reaction is shown, and that leads to obviously allergy. For example, asthma. And when the inflammatory response is inappropriately directed against the host tissue, then it leads to autoimmune diseases. So in this table over here, few examples are shown. For example, in disorders that is acute, acute disorders include acute respiratory distress syndrome due to neutrophils, acute transplant rejection, asthma, glomerulonephritis, and septic shock. Cytokines are involved. In lung abscess, neutrophils are there. And in chronic inflammation, arthritis, for example, lymphocytes, macrophages, antibodies, asthma, eosinophil Ig antibodies, atherosclerosis, macrophages, lymphocytes, chronic transplant rejection, lymphocyte cytokines, and in pulmonary fibrosis, macrophages and fibroblasts are seen. After this, there is another topic, that is defects in leukocyte function. The most common cause of defective inflammation are bone marrow suppression, that is caused by tumors or treatment with chemotherapy or radiations, and metabolic diseases such as diabetes. Though genetic disorders are there too, but they are rare. Causes are defects in leukocyte addition in LAD1 and in LAD2. In LAD1, there is defective synthesis of CD18 beta subunit of leukocyte integrin, and in LAD2, there is defect in fucose metabolism. The second cause can be defect in microbacterial activity, for example, chronic granulomatous disease. There can be defect in phagolysosome function, for example, Chidik-Hikashi syndrome. are in rare patients with defective host defenses have been shown to carry the mutation in toll-like receptor signaling pathway. And the fifth cause is that there can also be gain of function mutation in gene encoding the some components of inflammasome, one of which is called as cryopyrin, and they are responsible for rare but serious diseases called as cryopyrin associated periodic fever syndrome. Over here you can see that defect in leukocyte function are also discussed. We have also discussed the Chidik-Higashi syndrome, chronic granulomatous disease. As I told you, it will lead, there is a defective oxidative burst, X-linked, for example, phagocyte oxidase, membrane components. Defect in complement system, for example, C2, C4 deficiency, or C3 deficiency, or deficiency of complement regulatory protein. They can also lead to defect in leukocyte functions. So this is a summary that there can be defect in addition, for example, defect in ENP selectin sugar epitopes. As I told you about LAD1 and LAD2. Defects of chemotaxis or phagocytosis, for example, Chidik-Higashi syndrome. A defect of microbacidal activity that will lead to chronic granulomatous disease. There is a deficiency of NADPH oxidase that then need the superoxide. Therefore, no oxygen-dependent killing mechanism are there, and definitely leading to a defective leukocyte function. That is all about the outcomes of acute inflammation, leukocyte-induced injury, and defects in leukocyte function.